I'm Maria Wilson.
And I'm Danielle Mandikian.
And we are scientists. We love science.
Yeah, we do. So when we aren't doing it, the next best thing is to talk about science. And what's really awesome is that we're surrounded by some of the most brilliant minds in research.
We're going to step away from the labs today to talk to other scientists about the cool stuff they are thinking about, working on, and imagining.
As well as how some of these discoveries just might lead to new medicines. So grab your favorite drink, get ready to unlock your science brain, and join us for Two Scientists Walk Into a Bar.
The show for scientists, science geeks, and the people who love them.
We're talking to people a lot this season about AI and machine learning. So is there anything in your work that makes you very excited about AI or machine learning? Any caveats?
It's really taken the amount of work that we need to do in the lab to find hits and made that, a much more reasonable number.
This is great for us because we can use structural predictions to generate new hypotheses and then test them in the lab. Kind of iterative process of modeling and then testing really accelerates our work.
I'd have to say virtual screening takes away having to physically do the experiments, and you can just run it in the machine and come out the other end.
I think it can really accelerate drug discovery, so I think it's here to stay, so we should just embrace it.
Hey, welcome to the show. We're coming full circle. We kicked off this season with AI and machine learning and then talked to everyone that came on. So now, today, we are finishing off the season with another expert on AI and machine learning, Rich Bonneau. Welcome to the show, Rich.
Thanks for having me.
Let's start with the basics. Rich, can you please define for us what is machine learning and what is AI?
Well, for me, machine learning, it has different definitions. So we could start with the more mathematical definition. And so there you could say that machine learning can be algorithms that learn, meaning, in the sense of regression or earlier statistical methods, you take data and there's the key part of the algorithm ends up being some parameters that then give you the line that is how the mean will change as some other variables change. Machine learning, I think, is a much broader category that involves lots of nonlinear, multi-layer, much more powerful methods that can actually reason for different instances across bigger spaces of the data. And so machine learning, from a substantive perspective, are these algorithms that learn from data. And I think most of the time we're thinking of some of the most powerful algorithms that are implemented with neural networks and implemented so that they can be optimized on and trained on GPUs. So that's the sort of class of algorithms. The other way that you could define it is what these things do, the outputs. And there we could think of machine learning being something that predicts and something that can say, if I take some data, I can identify and label and predict and annotate. And that would be one kind of machine learning. And then there's generative AI, which means I see a bunch of stuff, I learn a model for the world, but now I can use that model to make new things that are wild extrapolations and combinations and compositions that didn't exist before. And so I think for the practical definition, we might separate it into generative AI and AI for annotation. But I think mathematically, that substantive definition, a lot of those methods end up being the same.
What are you most jazzed about at the moment?
Well,
That's super broad. I meant in the context of AI and machine learning.
Yeah. Well, I got a kitten for my birthday about 11 months ago, and that's still pretty hot ticket right there. She's a lot of fun. But you mean science-wise? Yeah.
I feel like it'd be really hard to model the actions of a kitten because they're so erratic. It would take me a while to figure out those rules.
There are no rules. Yeah, don't even try.
Pure chaos.
I think there's a couple things that I'm really excited about. But, one thing that I'm really excited about is that molecular design and generative AI for making things in the laboratory is starting to work.
So what do you mean by making things?
Well, therapeutic antibodies, small molecules that have a chance of having the right properties to become medicines one day. I mean, these are things that I'm really excited about. One is just the underlying structure and physics basis. And I think there's a huge amount of work going on right now all across the field where we're using machine learning to predict and design structures. We're using machine learning to encapsulate scoring functions and energy functions. There's sort of the rules for what's low and high energy, what should happen and what shouldn't happen, and which way the system should evolve if left to itself, right? And so all of that work, whether it's the core machinery of predicting structure or it's the scoring function and the more physics side of things, encapsulating all that stuff in neural networks seems to be just mind-blowingly efficient. There's lots of ways you can correct for the mistakes you make in weird ways by training with the data and also make them more accurate. That's like completely blowing my mind right now. And then the other sort of side of it is, well, there's also 1,000,001 things, well, let's say 10 things, that you want any given molecule to do, right? And so if it's material science, you might have interactions and durability and all these sorts of things that are properties of the whole system. If it's a medicine, the thing has to interact with an animal, and which has a whole range of complexities and compartments and other things it could bind to. And so taking those things and coming up with coarse-grained or really strongly predictive models that you can use to guide design, that's also something that's really, I think, quite exciting in a number of design fields. Beyond just what we do in biotech, I think there's a lot of places where that's really exciting.
Can you talk a little bit, I mean, just to kind of give context, because this isn't like my area, and I'm sure there's a lot of folks that just the thought of it seems kind of just massive.
Yeah.
Can you talk a little bit about designing therapeutics like with this type of technology? Like how does it change the speed, turnaround, or other factors that you think are really important?
Yeah. So let's imagine we have just an antibody in the laboratory and we want to make it bind its target in the human proteome stronger, right? So we have one big protein that is not going to change much because we evolve really slowly compared to bacteria or whatever we do in the lab, right? So it's not days, it's, generations,
Yeah.
So the human protein stays basically the same, and it's got some funky shape and charges, and it's cool looking, right? We want to bind a particular part on that, right? Well, the machine learning for that has gotten very powerful in a few different ways, but one is that whatever you do to improve that interface, you shouldn't break the fact that that molecule, the antibody, still also has to fold. So the machine learning, just one really, there's a lot of different facets to how we do that properly, but balancing the folding and the composition, the self-assembly of that molecule with its function, which is those loops that stick off the end of the antibody have to bind the right place on the target protein, that's something that machine learning does really well. And the way that we might model the interface is by thinking through the structure and the biophysics of the binding. And of course, it's encapsulated in machine learning for computational efficiency and for a number of other reasons. But then a lot of how we compose the sequence so that it folds into an antibody is just based on huge amounts of experience, huge numbers, millions and millions or more sequences of valid human antibodies pulled from repertoires of healthy people. And those sequences are used to train a model that just will always generate sequences that are antibody-like. And so we have one part of this machinery that's spitting out a tape in the way of like an old-style Turing machine, or a lot of models of computing. There's like a reader and a writer and a tape, and this is how you teach algorithms early on in computer science, in a person's career. Well, that's kind of what we're doing, right? There's the sequence, and you're predicting the next amino acid, and that sort of purely sequence-based machine learning model. Quite complex model, but still the representation is just a sequence. It can actually make things that fold pretty well, but then those sequences, once they're generated, can go into a model that makes further modifications using a totally different representation of the system that's more structural, more physically real, visualizable, all that kind of stuff. And so you can imagine different machine learning methods interacting, bouncing ideas back and forth. And so there's a bit of an ecosystem of these different codes that work together to do that. But that's another thing that machine learning does really well. It translates between different structured spaces, right? We use it to translate between images and text. We use it to say, give me this and generate some image. We use it to translate between human languages. You can talk to machine learning and write code, and those sorts of structured translations are done really well by machine learning. And we can use those to go between sequence, structure, and function space when we're thinking about proteins. So that's something that's really exciting there, is that these things that are really difficult translations that, 50 years of hard work in the lab has given us a beautiful amount of experience with, we can take that experience and encapsulate in something that can actually automatically and very quickly do those translations.
Yeah, and what it makes me think of too is like I sit on a very different side. I'm in the kind of development space, so it also seems very promising to me on the likelihood of the success of it going through all of these different stages that we'd want to carry it, because sometimes we'll get molecules that for so many reasons. Like we think it'll be fine, but there's been like, introduction of like charge patches or things that we know are going to wildly change the distribution in the body.
Yeah.
And so it's kind of cool to think about it from that perspective, because you take into account based on structures and sequences that have already been, successful, I guess you can say, and try to like avoid those design problems that pop up.
Yeah.
Because it's an amazing amount of time and money that goes into like fixing when things go wrong. Right?
Yeah, that's right.
Yeah.
So there, I think the design of antibodies is just a great example where lots of different types of machine learning technologies and approaches can be put together in an integrated pipeline. You can think about cleaning data and aligning data. You can think about actual generative AI. You can think about filters and safeties and multiple downstream properties. All of those different kind of considerations can be made to kind of interoperate. And it's not exactly a data standard, but it's a standard way of formulating problems that really does allow you to snap things together into a pipeline. So that's kind of cool. And then the other thing is, if you're designing stuff, you might consider billions of molecules.
It would be nice.
So whatever you're doing to say if they're good or not,
yeah.
It should be pretty fast, right? And machine learning these days, again, if we started off with those two definitions, like the mathematical and the practical. Mm-hmm. On the practical side, a lot of machine learning these days is implemented on really powerful computing hardware, things like GPUs. And so much of what we do, you spend a lot of time, you amateurize a bunch of costs training a model, but then you can use it, in milliseconds flat to do things that would otherwise be really complex simulations or computations. And so there's a lot of that where we do a computation once quite efficiently, and then the machine learning model is just an efficient way of remembering what you've already computed.
Hey, Danielle.
Hey, Karen.
Hey, Danielle.
Hey, Wellington.
I want to know more about neural networks. What can you tell us about that?
I'm still learning about all of this myself. The way I kind of think about it, at least at my level of understanding, is the neural networks are these kind of associations that are a lot more complex and kind of build off of each other.
Danielle, do you use neural networks in your work?
Totally. Whenever I tell you that I'm stalking cells, that's literally what I'm doing. I like to take these really complex fluorescent images of tissues, and I use these neural networks to actually segment out key biological features in my images. Another thing I wanted to ask you about is, how do you see AI and machine learning in the kind of discovery phase? So not just on the design of our therapeutics, but the targets that we would go after.
Yeah. So for target discovery, that's actually a really open-ended question, right? And so you could imagine finding ways of making assistants and tools that people use in essentially the same pipelines and processes, large pharma companies already use, smaller companies use. There's lots of different ways people decide what targets are going to work on. And those ways probably vary quite a bit across the industry. But if you had a machine learning model that had read all of the literature, all of patent lens, anything that has been publicly disclosed from any of the regulatory bodies, et cetera, well, just being able to ask that thing questions and have your answers generated in an efficient, kind of nicely summarized way the whole time, this is the minimum that we're going to see this process influence. But now we can imagine multimodal models that know about a little bit about how cells interact with each other, how genes interact with each other in cells, where things are expressed, all of these sorts of things, the degree to which different genes are expressed in unique patterns in some cells and not others. Imagine that cross-indexed with everything we know from everything that's been written. So you'd imagine that large language models actually could allow us to really start to reason over all the data that we have and really help the process of target discovery.
So when you say large language model, that means that it's able to interpret from different types of data sets, is that right?
Yeah.
Okay. That's really cool. It's hard for me to imagine.
Classes of models that can see essentially all the text relevant to a particular pharma company, all of the images, all of the literature, and all the relevant public information. These are the sorts of things like Llama and Gemini that you can train on everything, but you can also make those train to subsets of data. You can make those so that they've seen everything, but they've also seen sort of proprietary or data relevant to a particular industry, right? And once you have those models, you can use them in all kinds of ways. You can use them to help find data and organize data. You can use them to answer queries, but you can also sort of, instead of having them interact with humans, you can have them interact with other programs to sort of, in real time say, anytime I'm interested in something, I've got a targeting arm that says this will hit the tumor, what should the other arm be? Instead of asking a few questions and doing a little research for your favorite couple, why not just raster over the whole proteome there and get a ranking that's sort of unbiased? Who knows? But if you have criteria, if you have downstream processes that you can encapsulate in robust pieces of code or considerations or validation or metrics, well, then you can use these things to generate every possible combination of hypotheses and then put them through these downstream filters and things like that. So there's a lot of different ways you can use machine learning models for target prioritization and target discovery.
Let's take a step back. Can you tell us about one of the earliest kind of intersections between biology and machine learning? So
machine learning has had a really strong impact. If we stay close to home and think about biology, in 1997, there were machine learning methods that would, given the sequence of a protein, predict the secondary structure, how the backbone either folded into helices or strands or these different sort of primary shapes the biomolecule traces out as it folds. Those were really, really useful methods. And so as early as 97, we saw these sorts of things working. Now, these were cases where we were predicting three things, not helix or sheet, helix or sheet, right? There's three things you could be. Mm-hmm. And then we had an immense amount of training data to predict those three labels, right? The other thing is, you're pretty much an even balance of being those three things. So when you make your mistakes, there's also things like if you're a helix, you don't go helix sheet. So there are all kinds of rules for like how smooth these predictions need to be. And so in many regards, it was a really simple thing compared to what we're doing today. But it was really powerful, and it really made a big improvement in a lot of the downstream things we did with proteins at the time. I think machine learning has also been useful in lots of data capture, lots of informatics. There's a lot of machine learning built into DNA sequencers, the way in which these things are actually properly processed, the autofocus algorithms, all these crazy things on big machines. So there's a lot of like hidden ways that smaller, older machine learning methods have done quite a bit, even in the early days of sort of scaling up biology.
That's really cool. I don't know, maybe because I'm a social media delinquent. I'm not engaged very well, but it kind of feels like, people are talking about AI and machine learning so much more than they used to. So do you think what do you think's driving that? Do you think it's that there are certain advances that have happened more recently, or do you think it's a communication piece?
I think it's a mix of both. But one thing that's interesting is that if you do something very technical and it's in the shadows, that's really different than if you produce something that is commodity that people can play with, that people can have fun with. And the other thing is the advances when it comes to language, they're just so compelling because we are a big part of our day-to-day and a big part of how we interact with computers and the internet is through language. There's actually, I just heard a really wonderful lecture by Jan LeCun, who's quite active on social media discussing the relative risks of machine learning and things like that. And he's also a really early inventor of a lot of deep learning technology. Well, he was talking about how language isn't everything, right? A robot can't clean up the dishes after dinner, and he had a bunch of examples where there are simple things where the intelligence that's needed for that task goes beyond language. One of the things that's interesting is that vision and language we've seen huge progress in machine learning. And we found some edge cases where that kind of analysis of language is also useful for things like protein sequences or interpreting genomes or scheduling, certain events that happen in some transportation system. Well, how general is that? And how different are those really compelling commodity vision and language applications? How general are they to the huge range of tasks we need to do across multiple industries? That's kind of an active debate right now. But, on some levels it doesn't matter because it's so much fun to play with. Like my 13-year-old with DALL-E and ChatGPT can basically generate a comic book about our cat in 20 minutes, And it is pretty cool.
Yeah.
So.
Hey, Danielle, will you go a little bit deeper into these protein states?
Oh, yeah. I mean, that's pretty cool lingo to drop that's a helix, that's a sheet. All it is really talking about is imagine a protein is kind of like a pearl necklace. There's a bunch of amino acids strung together. Well, they don't just live as a single chain. They have these what we call secondary structures where they can fold up and make a spiral. That's a helix. Or they kind of organize into like a crimp kind of pattern, which is what a sheet is. And so all what he's talking about are different secondary structures that these protein sequences can actually form.
Danielle, do you use large language models in your work?
Not large enough. Do you have like a favorite story where you've gotten to like witness this come into play and really change the ability to treat a specific disease?
Right now, a lot of our favorite stories are on the molecular design side of things. Some of these things are, just improve affinity, improve these properties, remove these liabilities, and that's been a lot of fun. Some of them have also been revolving around, sensing the tumor microenvironment and getting proteins that sort of understand allostery or sense different components of the tumor microenvironment. On the large language model and target discovery sides of that, I think as an industry, we're still learning a lot about how that's actually going to play out. It takes a bit more time to validate those hypotheses, and so I don't know if we've really closed the loop on that application.
I think I'm still just fascinated by the thought of bringing in all this information about, biochemical pathways and how cells are interacting. I can't wait to see how that kind of evolves out because, one of the things that we struggle with, at the bench is even just picking, if I have question X about this, cancer immune scenario that I want to try to treat, even just finding, the right model that is mimicking what we would want to try to treat in the clinic can be very challenging. So I also really kind of look forward to, how this machine learning and information about the biological system could also inform how we would change the way we test things in vitro or in vivo.
Experimental design.
Yeah, because it's in. Yeah, I mean that's always been the thing I keep waiting for because this isn't my shtick, but I think it's really dope and I can't wait to see how things play out. But a lot of stuff for studying immune engagers or things like that, it's really tricky to even interpret the data for just understanding all the nuances of those preclinical models, what actually works, what doesn't work, and why. And it'd be so cool to see how machine learning can really transform that space for us at the bench.
And I guess in those settings, it's not just particular perturbations or things you can try, but it's the combinations of those things with other things and other things. And so you can't really ever exhaustively enumerate or just say we're gonna screen every possible combination and find the best, safest, most efficacious route to go here. You actually do need to design the experiment. So this is a great question. And one of the things that happens in machine learning is we take something that's existed for 50 years in statistics and we give it a new name, and we call it machine learning, right? And so it's really awesome, and it helps your citation count. There's so many advantages.
Good to know.
I love doing it. Yeah, it's really wonderful. But there is a really active field of error estimation in machine learning. So if you have these really complex models, how do you even put error bars on things? Mm-hmm. And this is going to get to your question. It's a little bit of an indirect route,
but I promise. You're singing
the
song of my people, yeah.
So how do you estimate error in these really complex models? Also, how do you decide if I get some new data point, how much will it improve the model? Right? The model's built to say how likely is that to be the right thing. But what if you also said, well, how new is this for you? Have you seen anything like this? Is this excitingly new, right? And so can you, as you're designing experiments, take shots on goal and try to get things that work, but also don't test too many similar things and don't test too many things that are so close to training examples that they're trivial and they wouldn't really improve the model. And when you put a lot of those things together, and error estimation is part of the machinery that you need to do this, you get to something called active learning. And this is the idea that you're not going to guess. You're not going to try to get the answer right in one go. You're going to say, I have a certain number of iterations that are in the budget, and I'm going to get there on purpose, and I'm definitely going to get there. And the way to do that is by iterating this experimental design, update the model, test your next things. But when you test your next things, you use the model in a very careful way to design sort of this optimal combination of improving the model and exploiting the model. And this is when we hear lab in the loop. Mm-hmm. This is, I think, what people mean when we talk about integrating experimental design and machine learning, or when we talk about sort of optimal uses of machine learning. Often it comes down to, again, you're in this high dimensional space, you can't test everything. The experiments, some might be very costly, some might be very cheap. How do you put it all together into an overall protocol and get to the right answer as fast as possible? It's quite complex.
Yeah.
But this is like the best question you can ask, because all of these things are working to different degrees. But when we try to use them in a complex environment, we want to make something that goes into an animal and makes perturbation that alleviates a disease. There's so many layers to getting that right that you can't test everything, and you need decision-making frameworks like that.
Hey Danielle, Rich was referencing the lab in a loop. I know that's come up in a previous episode. Can you remind us what we should know about that?
I feel like lab in the loop would, if scientists were to get tattoos, that would be the tattoo of 2024. Honestly, because everyone's talking about it. Basically, lab in the loop means that you are kind of bridging what you do at the bench with what you can do in theory by computation. And so like machine learning and all of this is really great for understanding data, making interpretations, linking things together, but there still have to be assumptions that are checked in the lab. And so that's really what this is this kind of like, fluid movement between both types of research that integrate together. And so like think about some of the stuff that we do with like the organoid work and thinking about how could this be interpreted, or how could data coming out of these organoids be used for understanding complex biological processes and then making predictions based off of that. That's a fantastic example of lab in the loop. So out of that body of work, where's your heart at and your most excitement?
So for us, we're really focused on generative AI, making new things. And by us, I mean myself and my multiple personalities. Okay, now I can't say we with a straight face.
Yeah.
We're really excited by generative AI. And so what we need from those methods is for them to take risks, for them to produce diverse ensembles, for them to understand dynamics a bit better than some of those early methods. And we also need them to be blindingly fast, because if you're doing generative AI, you're going to make billions of things and narrow them down and filter them and have multiple considerations and multiple properties you're optimizing. So we're speed demons. That's one thing. And we also need to make sure that when we're designing, we're not stuck to what evolution found before, right? So most of what happens in biotechnology revolves around the IgG fold, the antibody, our favorite way of conferring function and affinity, all these things, right? Well, there's so much evolutionary information surrounding that protein family that if you predict the structure of the antibody, well, the loop, you just made that loop. It's a brand new thing. It hasn't existed before. It's been matured and mutated and all these things, and then it's sitting off this IgG fold, which is a dominant attractor on the evolutionary sequence landscape, right? So how do you do the right thing for the loop, but also kind of properly predict the easy part, the antibody fold? So we need to make sure that our methods are fast, but they also have to appreciate single sequence design. They have to look at a single sequence and not extrapolate the answer from evolutionary information. They have to cook up the answer properly for that particular sequence. And so that's a little bit different than a lot of the approaches that are most successful for natural proteins.
You could easily convince me to invest what little money I have in whatever you're doing. But where, how fast is it to actually get this to happen? Where is the hype or the common misconceptions about making this turn into the smooth story that we hear?
Well, yes, implementing this is easier said than done, right? So let's imagine the code works. Well, there's another algorithm that works pretty well, which is just immunizing animals and using essentially the algorithm built into the adaptive immune system. So our things work, but then there's some targets where the right thing to do is the experiment, and that's actually cheaper and better than doing the machine learning. There are other places where it's a tough target, and this sort of machine learning method is going to actually break new ground. And so now we have to sort of have a mix of biologists who aren't trained in computer science and computer scientists who are often woefully undertrained in biology. And somehow they have to get together and figure out when to use which of these 6 methods that we've been so kind of relaxed talking about. So this is the growing pains there are that we have to reform how a lot of these decisions are made in these molecular teams, these drug discovery teams, and that's taking a little bit of time to do properly. But it's fun, And people are having fun because things are different. It's also kind of terrifying because the field is moving so fast, right?
Yeah,
and so we
feel it.
we have to work for 2 years to kind of implement something, but the rug might get moved under our feet a teeny bit in that time, or a lot bit, or the rug might disappear. So
I mean, I really like that perspective, though, that it really needs to be that combination of two fields or two kind of like training backgrounds, because I feel like sometimes people hear the hype and they're like, oh, we want to totally move away from doing in vitro or bench work or whatever.
Yeah.
And I think it's really important to drive that message home that you need to like be working together and figure out like what's the information that we could actually generate to help support these models, and then we can also give feedback of where we're really, really struggling.
Yeah.
Right?
Yeah, that's the key thing, is that we have a really close relationship with the teams into the drug discovery side of things, and there we go back and forth quite a bit about, first of all, what tools are needed. Mm-hmm. And they're not always the tools that the machine learning people are most excited about, right? So we're very technology first, and that doesn't always work out. But, I think it's been a lot of fun in the sense that there's also a lot of high throughput experimental capability that the machine learning people just don't know anything about. And so sometimes the conversations go really well in the sense that we say, well, if we were going to train a model like that, we would need data that looks like XYZ. And then the biologist will just pat the machine learning person on the back and go, don't worry, little guy, we can collect that data by next week, And so, it goes both ways. Sometimes the things we want to do kind of break down and they're a little bit more impossible than we'd like. And other times that's just a pretty straightforward experiment. And the only reason you don't know about it is because you did your PhD in CS.
So, like, how does bias impact kind of like model building or interpretation? So where do you guys struggle with it?
Well, so we can think about bias in the societal way, but we can also think about bias in the broader way, which is your training set does it have bias in how it was collected that's then going to hurt you no matter what you do downstream? And I think when we think about what are the cohorts look like that come into our trials and things like that, ultimately that's also the substantive reason why that societal bias can be dangerous, because if we don't have an accurate representation of human genetics, then we're not going to make the best predictions, we're not going to make the best medicines. And I think for us, we have to be really careful about what goes into these training sets, and it comes back to, I think, some really tough unresolved questions in active learning and experimental design, right? If you're designing these experiments and going back and forth, how do you also make sure that you're not leaving a whole swath of space unexplored? And that could be composition of cohorts, that could be high throughput experimental design, that could be what data sets do we really focus on cleaning up and ingesting. There's lots of different ways we have to fight that sort of data bias question. And then once we get to the large language model and some of these other things we discussed, target discovery, target prioritization, tasks where you ingest every piece of text of 125 years and then you hope that you can get something out of it. Well, there I think there is actually quite a bit of different styles, different societal biases, different things that are probably embedded in all of that text. And so there we're probably going to have to be very careful when we use those methods. I think the key is finding metrics that would also give you some sort of an indication about how critical the bias in the output and the training sets are, right? Can you find a way to operationalize some of the questions that surround bias and then test them to really make sure that the model is performing the way you think it is? I think that's the key there. And so for molecular stuff, we can think about that, and we can do that. For some of the more text-based stuff, things that like reach the more business-facing parts of the application of the large language model, I think we're just getting started there.
Yeah. Again, that's all really cool. And again, I'm super excited about thinking about identifying that bias in our preclinical models, because it's there's so many parallels to this, like that are like easy to conceive, such as like sampling from different populations and that kind of bias. But there's so much that I think we take for granted, even on the models that we're trying to train our basic biology understanding from. What do you think is going to be the coolest thing that comes out of this? Like, what would you like to see pop up in 25 years if everything goes as smoothly as we like stories to go?
All the rate limiters of today, it's not machine learning, it's just science. The machine learning, or the rate limiting steps of today are not, the rate limiting steps of tomorrow. They're the kits you can order. They're the things that just work, right? So if we think about that, machine learning is a big part of how we're going to make these advancements, but there's all kinds of things contributing, right? I think we're going to be able to think about medicines that actually have more valence, have more different perturbations that they make in combination that essentially can sense what's happening in the cell. So things that can sense, things that combine multiple targets, things that are actually more complex medicines. I think that's something that if we make it routine to approach a single target, then we're just going to unlock this possibility of actually thinking through combinations that are more complex, which will then open up all these questions you were asking and talking about modeling the cell, which is more complex still and still quite unsolved. And so we're going to reveal the next layer of complexity, which I think will be these sort of multiple target approaches.
So moving from the if-then statements to the if-then medications in real time.
Perhaps, yeah. And again, this is conjecture. I don't
like
really
conjecture.
know, but
That's
I like thinking about that and about tuning the cell with more than just a few perturbations, right?
I listened to something over the weekend, and I don't ski, but someone was talking to me.
Super dangerous,
right?
Yeah.
But for the people that do ski and survive,
yeah,
one thing that's really key difference between them and like me is like if I were to ski, I would be like, don't look at the tree, don't look at the tree, don't look at the tree, avoid the tree, avoid the tree. And like, all I would see are trees. and they were promoting this idea of like, people that successfully ski always say, follow the path, and they're always constantly looking through that way through. And I like the conjecture because that's, I think, is like the way to success, right?
Choose your route. Yeah.
You said you're a speed demon, right? And so what is the speed of all of this happening?
Well, so there are a lot of enabling technologies that transcend what we're doing in biotech, right? So there are enabling technologies that help us write better code faster using machine learning. So there's machine learning for writing code that does machine learning. That's pretty far out. A lot of the things that we do with machine learning, there are also algorithms that really effectively compress, store, organize, and can be used to search to find the right data. And again, organizing and feeding these models the right data is a big part of making them better. And so there are a lot of enabling technologies that actually make it better and faster to make the next version of these kinds of algorithms, including the algorithms that are the enabling technologies. So there is a bit of a loop that I think is continuing to increase in velocity. There's a limit to what people can keep up with. I think we're operating this system still, and so I think there's a limit. But I think things are speeding up a little bit. So I'll be very interested in seeing to what degree we can make good scientific use of ways of doing machine learning with machine learning. I think that's actually kind of interesting.
Yeah, it might be kind of a neat, in the end, social experiment, right? Because these technologies are getting faster and faster and faster, and it's kind of interesting to see, like, in institutions, how fast can we start to adapt and implement. Thank you so much, Rich, for coming. This was very interesting and really cool. Definitely mind-opening.
Yeah, and thanks for having me. This is a really fun discussion.
Danielle, I feel like sometimes AI and machine learning seems like such a big black box. Like, not everyone understands what it means, how it is actually applied in a specific field. So this is a great episode. Thank you to you and Rich for enlightening us.
I totally feel the same way. Like, even though like I've started using some of this stuff myself, it still feels like this really massive space with a lot of areas that we could explore. And I think that's kind of the cool thing. It's like knowing that you've got this like giant expanse in front of you and all the possibilities. I mean, that's what makes science exciting.
My question is for both of you, which is, what is it like to look at science with this coming on? It's very different than I think your training was, which was not that long ago. It was like last year.
Right? I wish.
But how do you think how do you see science changing?
I mean, fast, like you're drinking from a water, like a fire hydrant, but that's what's cool about it, right?
Yeah, it's like when you were dealing with dial-up when you're younger, and then you got the internet, and now there's like AI is talking to you all the time. It's crazy.
So Danielle, I have a big question for you then, which is, the pipeline to get a medicine from a molecule is so long. Are all these things going to actually have an impact on the time it takes to, do everything you need to do to test a molecule to bring it to patients?
I mean, I think time will actually tell. I argue about this sometimes with my friends. Like, this is something we actually talk about over drinks when we're at the bar, right? Is how long does it take to invest to develop these technologies, but then what's the output going to be? And that's the thing that's sometimes really hard to predict. You don't know how long it's going to take to develop a tool until you're at the very end of it. But you do know that once you have that tool in place, you can imagine all of the places where it really speeds up the time from bench to bedside. I mean, that's critical. Even if we don't know the answer, I think we owe it to ourselves and humanity to push and find out. And that's our show. Thanks so much for listening. If you haven't already, rate our podcast wherever you listen. It'll help new people find us. And make sure to subscribe. If you have any questions about the show, you can contact us at podcast@gene.com. And now for me, it's back to stocking cells.